So we're going to move on to Jonathan Marshall from Massey. Jonathan's going to be presenting to us on getting lost in the forest, the pitfalls of high-dimensional genomic data in random forest classifiers. Thanks, Jonathan.
Thank you. So yes, it's always nice talking later in the day because you can refer back to other talks. And so I appreciated Graham mentioning earlier that predictive AI, which is this is very much about, right? It's a classification problem, is old school. And which is kind of correct. And I think in passing he said something about that, perhaps generative AI has some problems with hallucination. And if we have time, maybe we'll make a random forest hallucinate. Okay, so this is work mostly by Helen Smith, who's a PhD student working at Massey University. And so the problem sort of arose, came in this context of source attribution, which Petra mentioned earlier. She did do a PhD in source attribution, which is where do people get campylobacteriosis from, right? So it's a foodborne illness. It typically, in New Zealand, comes from one of these three animals, and the scaling of the size of the animal might give you some suggestion as to where it tends to come from. Traditionally, this was done basically by genotyping all the things in the old school way, where you basically look at tiny little bits of 7 genes. These are housekeeping genes that are relatively conserved under evolution. And you basically just look at, do I have a new version of that gene, right? And when you have a new version, you give it a new number, and then you have a look at you've got a profile, a allelic profile there, and you basically just match them up, right? So you look at your human cases, and you see that first human case is similar, but not the same as one of the other cases and the first poultry case. And the second human case is the same as one of the ruminant cases, so that you can kind of figure out where people get things from. Now we're a bit more sophisticated in that it's way cheaper to just whole genome sequence the whole thing, right? And so we get 1,000 genes or 1,300 genes. But we basically do the same thing in that we assign an allele to each of those genes based on is it a new thing or not, right? The problem, of course, when you sequence everything is then different. So you can't just go, is this the same as the thing I've seen before? Because it's not. Everything you see is new, usually. But it's still a simple classification problem. So you might think, for example, that some genes are better than others at doing source attribution. Turns out that they're not. So the MLST genes are the ones on the left here. That gives you some attribution. The chicken is the dark red colour. And if we just pick genes at random and do the same thing, so just random genes around the genome, you get basically the same answer, right? So what's happening here is all the genes are basically correlated with each other, and so when one changes, another one tends to change, and so you get similar signals from all of them. Okay, but this is very much an old school problem. It's classification, right? We've got lots and lots of predictors, which are all the genes. We've got a simple outcome variable, which is chicken, cows, or sheep, or what have you, and they're all correlated, right? So perfect problem for predictive AI. Something like a random forest would be ideal, right? It can deal with high-dimensional data, lots of predictors that are correlated with each other. So you could just go and sequence a bunch of Campylobacter isolates from sources. That would be your training data. You'd throw it at a forest. It would train the forest up, and then you would then predict on the human cases, and that will tell you which source each human case likely came from. And you can implement this in a couple of lines of code. It runs in 5 seconds and gives what looks like a sensible answer, and that it sort of agrees with what we know already. However, this cannot possibly be good. And it's due to 2 things. The first thing is that our predictive variables are categorical, right? Because they're versions of genes. They're not numbers. Well, they are numbers. That's how we store them. But they shouldn't be treated as numbers. And treating them as numbers will give you a very bad answer. In fact, it will help you cheat. But that's for another talk. Anyway. Instead, they're basically categories, right? And we have a huge number of categories for each gene because there's lots of different versions of those genes. So on the median number of alleles that we see at our relatively small data set is 48, so 48 different categories. And the problem with that is to build a tree, you've got to decide which of those 48 go left at any branching point in a tree and which go right. And there's 2 to the 47 ways of doing that, which is a quadrillion, okay? So on my computer at least, it shouldn't take 5 seconds, right? So something else is going on. The other thing that's a problem is that everything is different, and whenever we sequence a new isolate of Campylobacter, we get something different again. So this is a visualization of our human cases. Each row is a human case, each column is a gene, and there's a dot if it's new, if we haven't seen that particular version of that gene before. So you can see that some humans have very new Campylobacter, and that there's lots of sort of horizontal stripes there, so they look different to quite different. And there's a few vertical stripes there which will be hypervariable genes. But the point is that just that essentially every human you see has something that you haven't seen before. And we've got thousands of these things, and the model shouldn't know what to do with them, right? Because all we've said is we've just got something new that we haven't seen before, and so the model can just shrug and go, I don't know what to do. Of course, it gives us an answer anyway. And what's going on is it's encoding those categorical variables into numbers, or different numbers than they already are, I guess. And the particular piece of software that I showed you before, which is Ranger, which is a very popular implementation of random forests in R, does it by target encoding. This is the default option. You have to read the help to figure out that it's doing this. And how it works is it basically takes your categorical variable, which in this case is a gene with the different alleles, which are our rows there, alleles 1 to 7. And you just basically find out what proportion of that allele is in source A, B, or C, and you put that into your table. And then you notice that allele 1 and allele 2 kind of look the same, right? The profiles look similar, and maybe allele 3 is similar as well, whereas alleles 4, 6, and 7 are quite different to those ones. And so what happens is you PCA that, you do a principal components analysis, and that allows you to see that there's an order to those alleles, and that number 1's up the top, and number 4's down the bottom, and something's in between. And that makes things a lot more efficient, because if you force you replace your categorical variable with this ordered thing, then to figure out who goes left and right, you only have to sort of chop it in between each one. Okay, so you go from 2 to the k down to k. Yay. So that's why it's so fast to fit. So now we're going to interrogate whether it actually does any good. And it doesn't do fantastically, which is mostly because Campylobacter, the types you see on Campylobacter go everywhere, essentially. But it does okay, you can see, for poultry. So this is true poultry, this is true sheep, this is true beef, and then the ones are what it's assigned. So you can see that most of the poultry are assigned to poultry. Most of the sheep are assigned to sheep, but it's not quite as good. There's a bit of confusion with beef. And beef is not doing as well. I mean, most beef are assigned to beef still, but sheep is coming there. But that's for the isolates for which there's nothing new. What about for the isolates for which there's new stuff we haven't seen before? How well do they do? Well, the model shouldn't know much about these things, yet poultry still does really well, which is a little bit concerning. The others don't do so well, which is perhaps what I might have expected. But the weirdness comes when we say, well, when we fit this model, we had poultry as the first level of the outcome factor. So in R or any software really, you've got a grouping variable, you've got to have an order to it, right? Now in this case, the order doesn't matter and shouldn't matter, but when I change the order, we get a different answer. Okay, so now our poultry isolates aren't being assigned to poultry, they're more likely to be assigned to beef or sheep, and we're doing slightly better on the sheep. It's like, what's going on here? That shouldn't happen, right? Changing the labels on something shouldn't give you a different answer, but it does. And so what's going on is that those new things that we haven't seen before haven't been ordered, right? They haven't been target encoded, which is what was happening. And what's happened is they've just been slotted at the end of that table. But the trouble is that that table was ordered based on association with a source. So they've automatically been associated with the first source. So that's why the poultry ones did so well, because we had poultry as the first source. And why we switch it round, the other ones do so well. Okay, so there's a bias that's inbuilt into that implementation of random forests, which is going to happen whenever this crops up. We can fix it though, which is good, just by basically slotting them into the middle of the table is how we fix it, essentially. So we can do some maths that shows that if you assume that you don't know anything, then they should be in the middle of the table, okay? And then you get the same pattern. So it doesn't matter, order doesn't matter. Shuffle the outcome variable and you get the same result, which is what you should get anyway. Okay, so we can fix that problem. We can get rid of that bias by being a bit more careful about how we do our encoding. But there's something else that goes wrong as well. In fact, there's multiple things, but I've only got time for this. And that's because this target encoding happens before we bag. So a random forest is built by basically resampling the data so that each tree sees a different version of the data, right? So that the trees are nice and different. That's the main bonus of a random forest, is you explore your data in different ways, then you average across them to reduce the variance, right? And so each of those trees sees a different subset, and they also see the things they don't see as the out-of-bag sample, which they then use to get the out-of-bag classification and the out-of-bag error rate. So one of the great things with random forests is that when you fit them, they will give you a supposedly unbiased measure of prediction accuracy. Now the problem here is that we encode before we split. So what happens is that the predictors are encoding information about the outcome variable in the out-of-bag samples, okay? So the out-of-bag prediction is easier than it should be, and so when the forest tells you that it's getting 90% correct, that's wrong. In fact, it could be very wrong. So this is pure random noise, right? So we generate a bunch of pure random categorical data. We then assign sources randomly to those categorical data. We fit a forest, and we ask it, what's the out-of-bag accuracy? So we've got 3 sources, so it should be 1 and 3, right? Because it's pure random noise. But in fact, it tells us that as we increase the number of categories, it'll tell us that it gets arbitrarily close to 1. So it thinks it's getting 100% correct, when of course it can't possibly, it's pure random data. So again, we can fix this. Don't use out-of-bag, right, is the easy solution, okay? So in summary, hopefully this has been a little bit of a maybe sort of step back and think about or get some detail about what the machine learning algorithm is actually doing, because it depends on both the package you use. We've looked further than just Ranger, of course. Just about every one that we've looked at has some issue. Don't assume that the package is doing the right thing or that maybe you're using it correctly. And always check your performance on an independent data set, right? That should be the default. So the paper's here, it was accepted yesterday, hooray. And I'll be happy to take questions if there's time.
Thank you. We have actually got time for one question.
Awesome.
If you would like to pose one, or anyone online.
No, it's easy.
No.
Excellent.
Please thank Jonathan.